For our next unit, we are going to be learning about cell division, which will include two processes, mitosis and meiosis. We'll be learning about the cell cycle, and we'll also be talking about something called karyotypes. But before we get into any of that, there are some words that we need to make sure we know. The first being DNA, a word we should already know. It's a nucleic acid that carries directions to make proteins. On DNA, there are certain segments called genes. These are the actual parts of the DNA that are coding for the proteins, which means when DNA undergoes transcription, these are the small parts of the DNA that are actually getting transcribed and then used to make a protein. There are different versions of these genes called alleles. So you could have a gene for eye color. It'll come in different varieties. Maybe your mom has a gene for brown eyes. We would say that's the brown eye allele. And your other parent has the allele for blue eyes. So different genes come in different versions, and we call those alleles. DNA comes in two different forms. So it's still DNA, but it's just kind of what shape is it taking on? So if it's in its very long, stretched out, stringy form like spaghetti, we call that chromatin. And if the DNA is really tightly packed up into an X shape, we call that a chromosome. When we look at a chromosome, it has two halves. So in this picture here on the right, you can see the X-shaped chromosome, and it's made out of two halves that we call a chromatid. These chromatids are connected at a point that we call the centromere. It's like the center of the X. Another term to be aware of is locus. This is just the location of an allele on a chromosome. So the other little picture here on the screen, we have a pink allele and a yellow allele. They are the same type of gene. They're just coming in different versions. And that is their location on the chromosome. You can see there towards the bottom. That's the locus of this gene. A karyotype, this is what we're going to focus on today. This is an image of an organism's chromosomes. So you can actually get all the chromosomes of an organism from one of their cells, and you can take a picture of them, line up the chromosomes, and you can figure out some information about this living thing. The sex chromosomes, these are the chromosomes that will determine whether or not the living thing is a female or a male. And for many living organisms, this will mean that the female will have two X chromosomes and the male will have an X and a Y chromosome. And we'll get some practice with this in our activity today. Another type of chromosome will be referred to as autosomes. These are the chromosomes that contain genes that control all of the other traits of an organism. So they don't determine whether or not the living thing is a female or male. It just has information, a bunch of genes that are coding for many other proteins that determine other traits. So here's an example here. It says humans have pairs of chromosomes. Look at the human karyotype to the right. How many pairs of chromosomes do humans have? Well, it looks like we have 22 pairs that are autosomes, the first 22 pairs. And then at the very end there, we see the X and the Y. So we have two X chromosomes here. And that's going to count as that 23rd pair. So in total, humans have 23 pairs of chromosomes, which gives us a total of 46 chromosomes. Chromosomes can be used for determining the sex of an organism, so whether it's a male or female. It can also be used to determine if there are any extra chromosomes or missing chromosomes, which would be chromosomal abnormalities or mutations. So in these two pictures here, you can see that the one on the left, that human would be a boy, a male. There's an X and a Y there. And then the karyotype on the right, there are two X chromosomes, like our previous example. So that would be a female. So speaking of these chromosomal abnormalities, you do need to be aware of a few of these. So these first two have to do with the total number of chromosomes. So if somebody has an extra chromosome, So normally there's 2 of all of them. If there's an extra one, so 3 of any of them, that's called trisomy. So tri means 3. There's an example on the slide here actually. So the bottom right example, if there's 3 chromosome 21s, we say that's trisomy 21. If somebody is missing a chromosome for any of the pairs, we call that monosomy. Mono means 1. So if there's supposed to be 2 and there's only 1 chromosome there, they're missing one, that's monosomy. Both of these mistakes in the chromosome numbers happen because of something called non-disjunction. And this says when chromosomes do not separate correctly, which happens during cell division. When you're making new cells, the chromosomes are supposed to divide evenly, but if they don't, that leads to the incorrect number getting placed into the new cells, which could lead to either monosomy, missing a chromosome, or trisomy, when you have an extra chromosome. Okay, last 2 mutations here, chromosomal mutations. So for a deletion, this is when a segment of a chromosome is removed. So you are completely missing a chunk of a chromosome. That's a lot of DNA that's missing. So we can see that in A in this diagram. We are just completely removing section D from the chromosome. And then duplication is when a segment of a chromosome is doubled, or there's just some extra bit of that chromosome that's added in. So for B, we can see that sections B and C, maybe it's gene B and C, they are just doubled. They're copied, which is making that chromosome much longer than it's supposed to be. Has too much genetic information in it, which could lead to some issues. Here's another example of a karyotype. So this is a dog karyotype. Dogs are living, which means they have DNA. If the DNA is tightly packed up into those chromosomes, then we can arrange this karyotype and analyze the chromosomes of the dog. And this question asks, how many pairs of chromosomes do dogs have? They have a lot more than humans. So the number of chromosomes that an organism has doesn't determine the size or complexity of the organism. So this dog has a lot more than us. Dogs are cool though. They should have more DNA, right? So it looks like they have 38 autosomes, but they also have that last pair of sex chromosomes similar to humans, so the X and the Y. That means that the total would be 39 pairs. It's a lot of DNA there, a lot of chromosomes. All right, I believe that was our last example. This concludes your notes for the karyotypes and just the introduction to this cell division unit. If you have any questions, please make sure you are asking. Thank you.